recently published a paper in Human Molecular Genetics which looked at how FAN1, which was a big hit on chromosome 15 in our genetic studies, might influence Huntington's disease, because FAN1 is classically known to be an endoexonuclease that resolves DNA repair, and in particular it resolves interstrand crosslinks, which are really deleterious to chromosomes. So they have to be resolved, usually during replication particularly, but at other times too. But it wasn't known that FAN1 had anything to do with Huntington's disease at all. However, we think that FAN1 does have something to do with Huntington's disease, because we showed that if you knock out FAN1 from a cell line containing long Huntington's repeats, that those repeats expand faster than when FAN1 is present. And we showed this across several different systems. So we are continuing to study FAN1 because we think that FAN1 and the mismatch repair machinery with which it interacts are likely to be very important in altering the onset of Huntington's disease. The problem is that although we have quite a good idea of how the mismatch repair machinery works, and we know that MLH1, which is part of the mismatch repair machinery, interacts with FAN1, we don't really know what they're doing at the repeats. And so we've been very interested in finding out, and we've been very interested since FAN1 has the opposite effect to the mismatch repair machinery. So if you knock down the mismatch repair machinery, you actually prevent the expansion of the repeats, whereas if you knock down FAN1, you precipitate the expansion of the repeats. So it looks like there's an interaction that's very important here, but we don't know whether that's a direct interaction or whether there's multiple mechanisms going on that are acting in different ways. So we're busy pursuing that at the moment. And the first thing that we've done, as you can see from the paper we published, is that we've looked for a number of different systems, cellular systems, in which to study that expansion. And actually that was really quite challenging, because the expansion had mostly been studied in human brains, so we knew it happened, and also in mice. But mice are not a system that are very amenable to quick, easy experiments. So we've been developing other assay systems with our colleagues in London, and we now have a number of parallel systems in which we can study this expansion. So what we're busy doing is editing in and out the different genetic variants that we found through our genetic studies in people, so that we can work out what the mechanism might be and what are its essential components. In my lab, as I say, we've been generating cell lines. We are also doing some mouse work, but that takes longer to come to fruition on various FAN1 variants. We're also very interested in repeats in the genome more widely, and we have a poster here in this meeting about that. So, a lot of the genome is formed of repeated sequences, and about 3% of the genome is what we would call short tandem repeats. And short tandem repeats are repeats like the one you see at the Huntington's locus. They have a repeating unit of between 2 and 6 base pairs, and they are repeated in the genome. And our issue with these is that when you sequence genomes, which you can do now very effectively, essentially the algorithms that call the sequence say, oh, this is a repeated sequence, I'm not going to read it and throw it out. So we've been very interested in this, and we are trying to read through those sequences. And I guess the other thing that we've been doing, which is to try and get a handle on the underlying mechanism by which the continued expansion of the repeat works, is we've been exome sequencing, so sequencing the coding sections of the genome in people with Huntington's disease who have very early or very late disease compared with that predicted by their CAG repeat length. And the reason for doing that is the assumption that those people are likely to be segregating variants of large effect, much more likely to be coding variants. So we are looking at coding variants and have detected some in the mismatch repair machinery and in FAN1, and we're currently putting those back into our assay systems to see if we can work out what's going on.